Hi everyone and welcome to this edition. My name is Sherif Abouidela. I'm a professor in the University of Sherbrooke, Department of Microbiology and Infectious Disease, and I'm also the coordinator of the RiboClub. And today it's my privilege and I'm honored to present two speakers from the RiboClub. For those of you who do not know the RiboClub, you can say it's an unofficial Army institute in Canada, grouping all RNA scientists in Canada, and many of you probably have heard of it or actually been part of it because it's also RNA. The RiboClub is also include many people from the States, especially the East Coast. Today we will have a talk by two long-term members of the RiboClub. The first talk will be presented by Francois Bachand. Francois actually started in the RiboClub as a student, and now he became a professor in the RiboClub. He got his PhD in McGill, University of McGill, working on polymerase assembly and regulation in the laboratory of Chantal Officier. He finished in 2002, where he actually went to do a postdoc in Pam Silver Lab in the Dana-Farber Cancer Institute. And then he came back to Sherbrooke. I mean, he actually not came back from Sherbrooke, he came back to Canada, let's say, to start his career in Sherbrooke in 2006. And he's since been in the Department of Biochemistry in Sherbrooke, where he actually started and established his independent research program with focus on gene regulation and ribosome biogenesis using both fission yeast and human cell culture. And he actually made amazing discovery, published a very interesting paper dealing with transcription termination, and taught us a lot about transcription termination in zombie, which is always an organism where we're seeing and discover new mechanisms that I would say sometimes which is going teetering between the human-like and the yeast-like type of mechanism. And he will present today one of his very interesting, I think, discoveries about different type of regulation of transcription and its relation to nutrition. So with that, I will let Francois present his talk. But before we start, just a few instructions. So I will be taking all the question by chat, so you can send me your question as the talk go in the chat. Send it to me. I will try to remove redundancy and put question together, and then I will read the question to Francois and he answer it. If we could not answer all the question, then Francois will be able to take answer the question later after during the other talk by chat, or contact the person directly to chat with him afterwards. As we go forward, I ask you as always to mute your microphone and also your camera if you're not talking or participating, to reduce the workload on the network. And if you have any issue, logistic issue, you can always chat with Leandro, which is his name is the RiboClub admin as on your screen, or you can send it to me. The name will be Abouidela on the screen as we go forward. So for now, I'm going to invite Francois to start his screen sharing and take it from there. Francois?
Thank you, Sherif. Yeah, so I'll just activate my share. Looks good. Just like confirm, Sherif, that you see the slides?
Yes. Yes, right. Everything is fine.
So, hello everyone. I'm very happy to be here from my tiny corner in the world with you today and represent the RiboClub, the Sherbrooke RiboClub, in this RNA collaborative seminar series. Today I'll present a new story that's very interesting and exciting to us, that links regulation of transcription speed with gene regulation via a mechanism that involves alternative polyadenylation. And the two main actors that are involved behind this story is a former grad student, Jan van Robeys, who is now in the US doing his PhD, and a very talented postdoc that's in the lab that came from Belgium, Carlo Jagesams. So before I start, I'd just like to give a brief historical perspective of RNA polymerase II transcription. So for those of you that don't know, the first RNA polymerase that was identified was in the 1960s from the bacteria E. coli. And then it took 9 years to resolve that the situation was much more complex in eukaryotes, with the existence of 3 different RNA polymerases, Pol1, Pol2, and Pol3, each transcribing a different type of RNAs. And through this 50 years of studying RNA polymerase II transcription, and thanks to in vitro transcription reconstitution assays and structural studies, we now have a reasonable understanding of RNA polymerase II transcription. So we know it's divided in 3 main steps, starting with initiation, where RNA polymerase II is recruited to a gene promoter, forming a pre-initiation complex that will be fired into elongation. And during elongation, nascent RNA is being synthesized through the addition of new nucleotides, and eventually this transcription event needs to be terminated. And we now understand that transcription termination is intimately coupled with 3' end processing via the recruitment of large cleavage and polylation complex, which includes many proteins, as well as proteins that we've identified, such as SEB1, which promotes RNA Pol2 pausing at the 3' end of genes. And within this cleavage and polyadenylation complex lies an endonuclease that will cut the nascent transcript and produce 2 different fragments. A 5' fragment that you can see here, that's going to get polyadenylated, and in the case of messenger RNA, it's going to be exported in the cytoplasm, and a 3' fragment that has an uncapped, unprotected 5' end, which is targeted by a conserved exonuclease complex, which includes XRN2 in humans, or RAT1 in yeast, that is thought to chase down polymerase and destabilize the RNA-DNA hybrid, promoting the disengagement of RNA Pol2 from chromatin. So this is known as the torpedo model of transcription termination, and there's strong evidence supporting this model. So now if we come back to transcription elongation, the textbook view of transcription elongation is a rather continuous process from which polymerase traverses the coding sequence after being initiating from a promoter. But now we understand that transcription elongation is not that continuous. It's rather intermittent, in fact, where there's a lot of pausing, arrest, and backtracking, which is the case of polymerase sliding back on the DNA template. And we further know that there is extensive variation in the speed of RNA polymerase II, both within gene and in between genes. And this is influenced by chromatin elongation factors. So what we know less, though, is what's the consequence of this? Is there any physiological or biological consequence, and does transcription speed influences gene expression? And if so, is it used naturally by cells to control different expression programs? So to begin to address this question, we used a previously described mutation that was previously shown to slow down RNA polymerase. And this mutation actually substitutes a highly conserved asparagine that you can see here to an aspartic acid residue. And this was discovered and identified by Malagon et al. And it was shown to slow down polymerase both in vitro and in vivo. So we used genome editing in fission yeast in collaboration with Renan Fernandez at Yale to engineer a yeast strain that would have in fission yeast, or one of our model organisms is Espombi, that would have this mutation in the endogenous RBP1 allele, which is the largest, which encodes the largest subunit of RNA polymerase II. So we selected different clones, and to look at gene expression, obviously we used RNA-seq, and we did these experiments in independent replicates using 2 different clones, and looked at the consequence of having a slow transcription rate on gene expression. The volcano plot that shows that you can see here show that we have identified hundreds of genes that are differentially expressed, both showing increased expression or decreased expression. And we also looked at splicing, as previous evidence indicated that the rate of transcription elongation actually affects the pre-mRNA splicing. So we use different tools to look at RNA splicing. And in our case, in our mutant, we did not find a global trend in either decrease or increase intron retention, indicating that this mutation, at least in our in the context of fission yeast, does not appear to globally affect RNA splicing. Looking at the functional gene classes that are affected by this slow mutation, in the downregulated genes, we found that genes that were enriched in coding in component of transmembrane transport, ribosome biogenesis, and iron assimilation. And for the upregulated genes, there was 2 major categories involving sexual reproduction in yeast, as well as in purine biosynthesis. And looking into deeper into the RNA sequence, what we found also is that we found 3 genes that encode an evolutionarily conserved core regulon for phosphate uptake in both budding and fission yeast. So these 3 genes are encoded by FO84, FO1, and TGP1. They are all activated by phosphate deprivation, and interestingly also, they are also negatively regulated by upstream long non-coding RNA. So we were curious to understand how a slow polymerase could result in gene activation. So we focused our attention on TGP1, as this gene was the most highly induced gene in the RNA Pol2 slow mutant. And TGP1 has been studied by other groups. As I said, it's activated by phosphate limitation through a zinc finger transcription factor called FO7, which activates the expression of TGP1. And as I said also, this gene is also repressed in phosphate-rich condition by an upstream long non-coding RNA, which has been called non-coding TGP1. And interestingly, this gene was identified as having 2 independent polyadylation sites, or a proximal poly A site and a distal poly A site. And the distal poly A site, in fact, directly overlaps with the TGP1 promoter. So it was proposed that upon a transcription of non-coding TGP1, the cleavage, the 3' end processing machinery would impede with FO7 recruitment and thereby repress TGP1 expression. But what's still unknown is how TGP1 is actually activated upon phosphate starvation. What's the mechanism underlying TGP1 induction in phosphate starvation still remains unclear. So we had previously mapped poly A site at the transcriptome-wide level in collaboration with Bin Tian group by his method known as 3' reads, which maps poly A site. And we looked at the data, and indeed, we found a strong poly A site at the end of the non-coding TGP1 locus, and several suboptimal or weaker polyA site in this non-coding RNA. And when we overlapped the RNA-seq data from our slow mutant over this 3' reads data, we could see, as you can see here, strong induction of TGP1. In red, there is their coverage, the RNA-seq coverage from the slow mutant. So you can see that there was a strong induction in the slow mutant, as I told you. But in contrast, in the long non-coding RNA, we actually see repression of the accumulation of the long non-coding RNA. And in fact, if we look at RNA-seq coverage and we segment it based on the polyA site, we actually see, and we calculate a ratio between the mutant and the wild type, we actually see a decrease in coverage as we go 5' to 3', suggesting that there's a decrease in the full-length long non-coding RNA in the RNA pol2 mutant. So this led us to propose that in the slow mutant, what could happen is that the weaker proximal polyA site would be favored relative to the distal site, leaving access to the Fo7 transcription factor to bind its promoter and activate TGP1 expression. So we wanted to test this hypothesis, and the first thing we did is to look at RNA polymerase 2 density across the locus. So we designed primers for 8 different regions to scan the locus by chip. And the first thing we did is RNA polymerase 2 chip assays. And the data is shown here. And you can see clearly, consistent with TGP1 activation, that in the slow mutant, we get more polymerase density in the coding region of TGP1, which are regions 6, 7, and 8. But in contrast, we see a decline in RNA polymerase 2 density downstream from the proximal polyA site, consistent with premature transcription termination in the RNA polymerase 2 mutant. So to get more evidence that transcription termination was ongoing, we looked at 3' end processing factors. As I told you in the introduction, transcription termination is coupled to 3' end processing. So we analyzed the density or the distribution of 2 different proteins, RNA14, which is a conserved component of the cleavage and polylation complex that will leave the CTD to bind a nascent RNA. And we also looked at SEB1, which is a CTD interacting protein that we've shown previously that promotes RNA pol2 pausing at the 3' end of genes. And as you can see here, for both RNA14 and SEB1, we clearly see a redistribution from the distal polyA site to the proximal polyA site in the RNA pol2 slow mutant. So this suggests that in the slow mutant, there's a redistribution of the 3' end processing machinery from the distal site to the proximal polyA site, and thereby we would expect this would give access or more access to Fo7 transcription factor. So we looked at this by chip. We tagged Fo7 in the wild type and the slow mutant, and sure enough, we saw a 4-fold increase in Fo7 binding at the TG1 promoter in the slow mutant. So this leaves us with a model where in wild type cells, where there is optimal transcription elongation speed, RNA pol2 will read through these weaker polyA sites in the non-coding TGP1 locus and will promote 3' end processing at the distal site and thereby impede with Fo7 recognition of the promoter and repress TGP1 expression. However, in the slow mutant, this slower elongation rate allows more opportunity to recognize these suboptimal weaker polyA site in the non-coding TGP1, and thereby this will lead to premature transcription termination and allowing Fo7 to bind its promoter and activate gene expression. So we wanted to get further evidence supporting this model, and we wanted to see whether actually premature termination was the direct cause of TGP1 activation. So what we did is to analyze the sequences surrounding the distal and proximal polya site. So here I'm showing you the sequence of the distal polya site above. You can see this AAUAAA hexamer, which is a consensus sequence that is also used in fish and yeast to recruit the 3' processing machinery, the cleavage site, and in orange, several SEB1 binding sites, which I've told you actually promotes RNA pol2 pausing at the 3' LMG. However, when we looked at the proximal, the major proximal polya site, the hexamer was non-consensus, and we had absolutely no SEB1 binding site and downstream of the cleavage site. So we thought that by strengthening this proximal polya site by the introduction of SEB1 binding site, we would propose RNA pol2 pausing and favor an proximal premature transcription termination in the long non-coding RNA, and thereby expect increased expression of TGP1. And this was exactly what we observed. You can see here, this is RT-qPCR relative to the wild type. We can see a strong induction of TGP1, and this is in the wild type RNA pol2. This is not in the slow mutant. So by pausing RNA pol2 at the proximal site, we get decreased levels of full-length non-coding TGP1 and increased expression of TGP1 expression. So this mechanism that we've seen, that we've described now in the slow mutant, where we can get explaining how TGP1 is de-repressed by premature transcription termination, we wanted to know whether was it applied as well to conditions of phosphate starvation. Is the mechanism that we've described in the slow mutant also the case in case of phosphate deprivation? So we planned an experiment where we grew our wild-type fish and yeast cells in phosphate-rich condition, and we split the culture in two, one that was maintained in phosphate-rich condition, and the other half that was grown in phosphate-depleted medium for 4 hours. And we looked at RNA pol2 density, RNA14 recruitment, and SEB1 by chip assays. So in the left column here is our data that I've showed you for the slow mutant, whereas on the right, I'm showing you the data that we got for the phosphate-depleted cells relative to the wild-type cells, the phosphate-rich medium. And as you can see here, again, in the phosphate-depleted medium, we got increased pol2 in the TGP1 locus, and we see a decline of RNA polymerase 2 downstream of the proximal polya site, as you can see here. Again, also for RNA14, as we saw for the slow mutant, we have a redistribution where we have increased binding of RNA14 at the proximal polya site in phosphate-deprived medium, and the same is true for SEB1. So apparently, the mechanism that we've described of premature transcription termination in the slow mutant is also the mechanism that appears to be involved in activation of TGP1 in case of phosphate starvation. So given this result, we wanted to see how widespread was the effect of the slow mutant on polya site selection. So we designed an algorithm, Carlo designed an algorithm, I didn't do much for that part, where he actually looked and the coverage between the different segments for genes that would have independent polya site. So based on genes that had that we've mapped previously with Bintian's group that had independent polya site, we mapped the coverage between for the wild type and the slow mutant, and then we calculated a ratio for this between the distal coverage relative to the proximal RNA-seq data. And in case of TGP1, as you can see here, we have a negative value, which is consistent with a preferential use of the proximal polya site in the slow mutant relative to the wild type. And so we found there was 2,200 genes that had 2 independent polya sites in fish and yeast. And out of these 2,200, we found over 500 genes that showed significant changes in the selection of polyasite. And as you can appreciate here, there was a very strong bias for genes that were using proximal polyasite versus genes that preferentially use distal polyasite. And you can see here that the non-coding GP1 is as expected in this category. Now, if we look at what type of genes are affected, either proximal or distal, so for the genes that that showed preferential use of the proximal site, out of these 441, 406 are protein-coding genes, and 35 are non-coding RNAs, including non-coding TGP1 here. And for the 406 genes, coding genes that were affected, most of them were affected in the 3' UTR, as you would expect, as most of these polyasites are in the 3' UTR. Here you can see an example where we have a gene with a proximal and a distal polyasite, and you can see that in the slow mutant, in red, you see a decreased coverage downstream of the proximal polyasite. So the next question that we add, so we have analyzed APA, alternative polylation, in the slow and the slow mutant, and we also analyze gene expression in the slow mutant. So is there any relationship between gene expression changes and alternative polylation? And in fact, we found that there was a significant overlap between the genes that showed premature or proximal preferential use of the proximal polyasite and genes that were upregulated. So there was 54 genes that overlapped between these two categories. And furthermore, there was a significant enrichment in these 54 genes for a gene involved in purine synthesis. So that was interesting. An example of this is this TIM11 gene, which encodes a subunit of the F1-FO ATP synthase. And you can see that in the exons of TIM11, you can see more coverage in red, more expression in the slow mutant, but you see reduced coverage downstream from the proximal polyasite. And importantly, we validated this by northern blotting. You can see that in the two independent clones of the slow mutant, we see preferential use of the short isoform, which is the proximal polyasite, and more mRNA abundance in the slow mutant. So if we sum up what I've told you so far, so using a mutant that was previously characterized to show, to behave or to slow down transcription elongation, we've seen that this causes major and widespread effect on polyasite selection. And this influence on alternative polylation actually resulted in the activation of specific gene programs, including genes that are involved in phosphate uptake, so the core-4 regulon, via a mechanism that actually prevents full-length or causes premature termination of upstream interfering long-coding RNA, and also increased expression of genes involved in purine synthesis through a mechanism that involves 3' UTR shortening. So what does it mean biologically? So what's the link between the phosphate intake pathway, purine synthesis, and transcription elongation rate? Well, it's well known that the phosphate uptake and purine synthesis have a direct consequence of the cellular nucleotide pool. Indeed, when you deplete cells from, or deplete the medium from inorganic phosphate, the first metabolite that drastically decreases is ATP, which is a nucleotide. And as ATP is an energy source to make other nucleotides, there's a drastic decrease in the nucleotide concentration when cells are in phosphate deprivation. And as you know as well, NTP is a substrate of transcription. And if you reduce the concentration of NTP, you actually slow down transcription elongation rate. So, and if you listen to my talk, I told you that reduction in transcription elongation rate can actually influence polyasite selection. And through this mechanism can result in the activation of specific programs of genes involved in phosphate uptake and purine synthesis. So what this led us to hypothesize is that genes that are actually important for the maintenance of nucleotide pools, such as genes involved in phosphate uptake and genes in purine synthesis, have evolved gene structures that appear to allow them to be responsive to reduced rate of transcription elongation. And we actually could test this experimentally by actually decreasing NTP concentration in cells. So using wild-type yeast, we can treat cells with 6-azouracil or mycophenolic acid, which interfere with the biosynthesis of NTPs, and have been shown to slow down transcription elongation. And in fact, when we treat cells with 6-AU and MPA, we get a strong activation of the core 4 regulon, as shown here, and we also get increased expression of genes involved in purine synthesis, such as TIM11, by preferential usage of the proximal polyase site. So what we think is going on is that RNA polymerase 2 actually can act as a sensor of nucleotide concentration in cells by controlling the rate of transcription elongation. And by doing so, can influence RNA processing event, as I've shown here for alternative polyadenylation. And alternative polyadenylation can have positive impact on gene expression, such as genes involved in phosphate uptake and purine synthesis to respond to this decreased levels of nutrient availability to get back to normal NTP concentration. So globally, we think our findings suggest that the transcription elongation rate can actually, in fact, control gene regulation or affect gene expression or specific gene expression program in response to nutrient availability. So again, I'd like to thank the people that contributed to this work, Carlo and Yann, with help from Marc Larochelle, help from Pierre-Etienne Jacques and Julien Béraud's lab at Yale, and the funding agency, and I'd like to thank you for attending today.
Thank you, Francois, for this very interesting talk. So I'm going to start taking your question, so please type them and I will read them. But while waiting for the question, I'm probably going to start by couple here, Francois, until the question come in. The first one is, you have shown us in the beginning that many of the ribosomal, many of the genes that are downregulated by the slow-mutant ribosomal protein genes, but then they did not come up when you talked about the polyadenylation, because they are downregulated for a different mechanism, or you did not look at them. They're normally short genes, so do they do something else?
Yeah, mechanistically, we haven't looked at them. We haven't focused much on the downregulated genes because initially that's what we expected. I mean, a slow mutant would maybe globally reduce mRNA synthesis. Clearly, that's not global because, I mean, there's not all genes appear to be downregulated, and there might be some RNA decay pathway that might compensate for this slowdown in transcription elongation, and that was shown by Patrick Kramer's lab years ago. But so we focused mostly on the genes that were upregulated because we thought this was interesting to mechanistically to get a gene activation by a slow polymerase. But yes, indeed, ribosomal protein genes are shorter. Not sure how this integrates and why they are preferentially reduced in the slow mutant. This may be linked to preferential decay or something that we might want to be looking into.
Yeah, so I start having some question coming in. One from Josh Zimmer. Do chromatin marks around the TGP-BP promoters change with a slow polymerase mutant?
That's a good question. We haven't looked at chromatin marks at the TG1 promoter. So whether repressive or obviously you would expect to see active mark. Now in the case of the slow mutant, as the transcription of the upstream non-coding RNA doesn't make it to the promoter, but we haven't looked exactly or directly at different histone marks in the wild type and the slow mutant. That would be interesting.
Okay, another question from Carla Knockbauer. Are there trans-acting factors involved in the rate-sensitive APA you described?
So just repeat the question.
So are there trans-acting factors involved in the rate-sensitive APA or sensitive polyadenylation you described?
We don't know. Clearly, in the only mutation that's involved in the slow mutant is the single residue that's being changed in RBP1. So it's possible that there's factors that are specific to the proximal polya site or that favor proximal polya site, but we think it's actually more a kinetic issue, such that, I mean, obviously, if you slow down the polymerase, at least for the proximal polya site, you can imagine that the slowing down the polymerase allows greater opportunity to recognize these proximal polya site, leading to cleavage and 3' processing. So we don't see a mechanism where there would be differential transacting factors favoring proximal versus distal, but rather a kinetic issue related to transcription.
Okay. So next question from Hubert Salvay, and he's actually asking, we know who it is. NC type 1 regulated at the level of transcription initiation by phosphate. So do you see actually a regulation at the level of initiation also?
Right. So we can exclude that it's happening, but when we looked at the level of polymerase at the promoter of this non-coding RNA in phosphate deplete or in phosphate-rich condition, or in the slow in the wild-type mutant, we see the same level of RNA polymerase 2. So we can exclude that there is some transcription contribution to the non-coding RNA, but at least in our experimental setup, we don't see any differences in RNA polymerase 2 density in the wild-type, in the mutant, or in phosphate-rich or replete condition.
So another very interesting question from Patty here, who's saying, well, interesting talk. Does a change in nucleotide production in pentose phosphate pathway resulting from different glucose levels affect transcription rates and gene expression? I think it's very relevant.
Yes, this is a very interesting question. And this is stuff that we're working right now. Look at different environmental conditions that would also be predicted to affect intracellular nucleotide concentration, and look whether we get different genes or the same set of genes are activated. So yes, this is a very important topic.
Okay. One final question here by Badal. Is a slow mutant phenotype sufficient for the premature termination and choosing proximal PA?
Is the slow mutant phenotype sufficient? Well, I think the experiment by strengthening the proximal site of TG in the wild type cells, I think provides very strong evidence that premature termination is sufficient to drive TG1 expression. So all together with our data in the slow mutant, we think that the mechanism of that we described here for TGP1 is based on controlling transcription elongation speed.
Okay, great. So thanks a lot, Francois.
Thank you,
exciting talk. Now we're going to switch to our next presentation. It's going to be presented by Andrew McMillan. So Andrew is also a long-term member of the RiboClub. He got his bachelor's degree from University of Toronto in biochemistry and chemistry, and then he did his PhD in Harvard from 87 to 92 in organic chemistry, and then he followed that by a postdoctoral work in the Center of Cancer Research at MIT in the lab of Phil Sharp, where he got interested in splicing, and he never actually got disinterested in splicing. This was kind of a change in his career, and he never looked back. And now, since 1990, from 92 to 96, he was assistant professor at the University of Toronto. From 96 to 2000, and then moved after that to University of Alberta, who is currently now a full professor in the Department of Biochemistry. And if you do not know Andrew, you would expect that he would talk about the mechanism of splicing. There's only few, I would say, four still left interesting in dissecting the mechanism of splicing, and Andrew, I think, is one of them. And I'm pretty sure that he will, if you did not have your fill of chemistry before, you will have it now. So with that, I would leave it to Andrew. Please share your screen.
Well, thanks very much, Sharif. And I'm really pleased to be with everybody away. This is a great series of meetings. So this is what I'm showing on this slide is my 16-year-old's version of spliceosome structure when he was 14 years ago. And it basically summarizes, I think, what we knew about the higher order structure of the spliceosome at that time. But we've come a long way since then. So as Sharif mentioned, my lab has been interested for many years in the mechanism of pre-mRNA splicing, nuclear pre-mRNA splicing that proceeds by the 2 transesterification steps shown at the top of the slide. And I'll focus for part of the talk on this first step in which the adenosine that carries out the first chemical step is selected from the intron by virtue of an interaction with the splicing machinery, an RNA in the splicing machinery, the U2 snRNA. And this reaction was first observed in human cells infected with adenovirus. So adenovirus splicing, which was the discovery of the intron exon structure. But as everyone is probably aware, the exact same chemical mechanism is recapitulated in the group 2 self-splicing RNAs, which are conserved from bacteria through fungi and plants. And in that case, the RNA itself folds into a structure that promotes these 2 reactions. But that's not what happens for spliceosomal nuclear RNAs. Just one second, what's going on here? Okay, yeah. So nuclear pre-mRNAs are processed by a machinery referred to as the spliceosome in homage to the ribosome. And the spliceosome is composed of particles that contain uridine-rich RNAs, the snRNAs, U1, U2, U4, U5, and U6. Those complex with some common proteins and specific proteins to form the so-called SNRP particles. Those assemble together to form the catalytic machinery. And on the right of the slide is shown, believe it or not, a purified spliceosome. And one can observe the assembly of the spliceosome in a number of ways. This is a non-denaturing gel in which an RNA is incubated in helinuclear extract, and you can see the time course that shows the formation of the successively larger complexes A, B, and C, with C being the catalytic spliceosome. Now, this is an old slide. It's a simplification, but it just dramatizes that assembly process, the stepwise association of the individual SNRP with the RNA, starting with what's referred to as the commitment complex through the A, B, and C complexes. And as I mentioned, C is the complex in which the 2 transesterification reactions occur. Now, for many, many years, a number of groups, a fair number of groups, have been interested in obviously studying the regulation of splicing, and have been interested in spliceosomal structure, but that study was complicated by this dynamic cycle, and it proved essentially impossible to purify the individual splicing complexes in a way that was amenable to, for example, X-ray crystallography. And so one was left with a dilemma, and the approach to that was the title of this talk, essentially the blind men looking at the elephant. And so that's the Indian parable where they're trying to describe this mysterious creature. And this is actually a beautiful Japanese print that you can see. Some of them are looking at the tusks, some of them are at the tail. These guys are on the top. And they're all reporting different features of this elephant, but that is an incomplete picture. So I'm one of the blind men who's been looking at the elephant for many years, and I'll tell you a little bit about our studies with a component of the U2 SNRP that recognizes this duplex that I spoke about a few moments ago. And then I'll also talk to you about a component of the U5 SNRP, PRP8, which is involved in the assembly pathway, but is also part of the core of the catalytic spliceosome. And these two proteins first came to my attention many, many years ago. This is a cross-linking experiment in which a chemically modified RNA is incubated in splicing extract, and over time you can perform the cross-linking experiment and immunoprecipitate spliceosomes, and you can see a sequential association of a variety of proteins with the branch region labeled here. And the two proteins that I just referred to are SF3B14, also called SF3B6, and PRP8, this large protein at the top. And so these are two of the snapshots or pictures that the blind men took of portions of the spliceosome, a portion of PRP8 on the left and P14, SF3B6 on the right. And I could have put up many, many structures here by many groups' attempts to understand the structure of the spliceosome by that sort of piecework. So bang. Oh, sorry. I'd be remiss, though, if I didn't show this structure, which was, until recently, the largest chunk of the spliceosome whose structure had been determined. This was work by Daniel Kermel in Kiyoshi Nagai's lab, which shows a large portion of the human U1 SNRP. And this structure took many years to determine by X-ray crystallography. So then, bang. A few years ago, people in the field were, I think, taken by surprise when the structure of an actual spliceosome was reported. And this is a complex isolated from Schizosaccharomyces plombi. And the structure was determined by Yi Gongxi's group in Beijing. And the structure determination was made possible because of several advancements. One was Melissa Moore's actually purification of, or protocol for purifying this type of complex out of POMB. But then the other was technical advances in electron microscopy that solved the problem of dynamics, because you no longer need homogeneous samples. This is a single particle method of analysis, and that gets away from the dynamics issue that I referred to earlier. So now we can review that cycle that I referred to earlier, and we now, in the last 5 years, have structures corresponding to all of the complexes that I show here. I think there are, what, 8 here? And this is even incomplete because there are additional complexes in this cycle which have been characterized. So we're getting a better view of the elephant, but if you just look at even this far away view, you can see that these structures differ considerably between each other, and that's a reflection of the dynamics of what's going on. So back to the blind people. We were interested in PRP8, the protein that I showed cross-linked in the slide a few moments ago, for a long time, and almost nothing was known about its structure until the last few years. And we focused in on a particular domain, and the reason why we cloned, purified and crystallized this domain was that mutant alleles had been identified in yeast that are suppressors of splice site defects. So you've got an RNA that won't splice, and mutants in PRP8 overcome that. And those correspond to what was referred to as domain 4. Excuse me. So we solved the X-ray structure shown here, and there are two forms in the crystal lattice, yellow and cyan. We'll call the yellow one the closed and the cyan the open form. And the first thing that struck us was that there's this conserved RNase H fold, excuse me, that forms the core of the protein. There's this hairpin, the presence of which had not allowed sequence analysis to identify this RH fold, just from looking at PRP8 structures. And in the lattice, that hairpin exists in this form, but also opens up, it's disrupted, and translated back in the other form. And in our original paper, we didn't really comment on that feature of the protein, but it turns out we believe to be fairly important. I mentioned that we chose to study this domain because of this concentration of suppressor alleles that map to this region of the protein, and those are shown as red balls in the space filling diagram. And you can see that most of them are located in that hairpin region. So we looked at this a little bit more closely over time, and what we were able to see, and this followed up on work by Magda Konarska and Charles Query, is that these alleles can be segregated into two groups. And what half of them do is encourage splicing of RNAs that are defective in the first step of splicing, and half of them are able to overcome defects that impede the second step of splicing. And we can analyze those mutants in a copper growth assay. I won't go into the details of that, but the growth of the yeast depends on splicing of a copper resistance gene. It's a well-known assay in the field, or by RT-PCR, as is shown on the right of the slide. And so, as I said, half of them overcome defects in the first step, half of them overcome defects in the second step, and we could actually correlate that structurally using X-ray methods. And so, for example, on the right, you can, on the left, you can see that several of these mutations actually stabilize the structure of the hairpin, but seem to have no effect on the open conformation. And on the right, we see stabilization of the open conformation, or actually right here, destabilization of the closed conformation. And so structurally, we're able to understand why there would be a bias between one of these forms and the other. And I've shown 4 examples here, but we have about 4 others that we've studied in the lab as well. And they can all be rationalized structurally. So here is a paper that came out of Nagai's lab after we published the structure of the RH domain, and it's most of PRP8. And so there's the RH domain. Here's a domain I'll talk about briefly called JAB-MPN, and that's involved in a step in splicing where U4, U6 snRNA is dissociated to form the U2, U6 snRNA structure that is the catalytic core of the spliceosome. And then there are vestigial reverse transcriptase and endonuclease domains here, and that's really interesting because that relates to functions in group 2 introns and provides an evolutionary link between group 2 introns and nuclear pre-mRNA introns. So we performed a sort of naive thought experiment. We superimposed our structures on this larger portion of PRP8, and that's shown here. This is just a thought experiment. So this is the closed, and this is the open conformation. And what we could see is that there were residues in this opened loop that were close to the JAB-MPN domain. forming a possible hydrogen bonding interaction. So we mutated those, classic experiment, and again in this copper growth assay, here's the wild type, grows up to a certain point in the presence of copper. We mutate the histidine shown on the previous slide. It actually grows a little better. Histidine to arginine, no change. The arginine on the JAB-MPN domain doesn't do as well. The double mutant, which is forming a salt bridge, a predicted salt bridge across these domains, grows really well. Okay, so let's go back to the elephant and consider a few things. What I'm just showing here is a superposition of just PRP8 from those structures that I showed you earlier. These are the EM structures. And in gray is the core of PRP8, which doesn't move that much, it jiggles. But colored are shown the transitions of the RH domain. And you can see there's considerable movement of this domain through this cycle. What is interesting is that all of these structures, and it's a little hard to see here, obviously, all of these correspond to the closed conformation. And so that open loop conformation is observed in none of these. And secondly, none of these structures show an interaction between the RH and JAB-MPN domain that I just gave evidence for you genetically. Okay. Oh, I should also mention that we did a study with Stephen Rader looking at a reduced spliceosome. And although this hairpin is very highly conserved throughout evolution, in reduced spliceosomes, it is not present or required for viability. Okay, so we'll leave that for a moment, and I'll talk briefly about SF3B. And SF3B is a component of the U2 SNRP that's involved early in spliceosome assembly, and I'm going to focus on the P14 component. So SF3B has 7 components. They're highly conserved evolutionarily from fungi through humans, although Cerevisiae does not have a copy of P14, but it's required for viability in human cells. We solved the structure many years ago. It consists of an RNA recognition motif, which is shown here, and actually that structure was solved between P14, shown in red and yellow, and a small peptide from SF3B1, which is the large component of SF3B. And what's not so apparent here is this gemisch of helices is occluding the canonical RNA binding phase. So we were confused as to what that meant. We did a series of experiments, cross-linking experiments, structural experiments, and some work with some model RNAs. And I think we were able to, at least for ourselves, convince us that the way that this interacts with RNA is that this bulged adenosine, this duplex between the intron and the U2S and RNA, that adenosine fits into a pocket on that surface. And that actually occludes the catalytic nucleophile for the first step. And so that interaction can only be transitory during the spliceosome assembly. With Reinhard Luhrmann, we located P14 within an EM structure of SF3B. And we were also able to perform a SAX analysis of RNA protein complexes, and both, and this especially is consistent with the model, the log and key model that I just proposed. So more recently, structures of the B complex from yeast in humans have come out. So here's yeast on the one side, humans on the other. And remember, I mentioned that P14 is not a component of the yeast SF3B. P14 is shown in red on in the human structure. And if we look at that more closely, what you see is that the P14 molecule is actually on the outside of the structure. So that contrasts with the EM modeling. It also contrasts with cross-linking studies of that were performed by Reinhard Luhrmann's lab with purified SF3B complexes, human SF3B complexes. And furthermore, this protein is about 50 angstroms away from the U2 duplex that actually interacts instead with SF3B1 nestled in here. So that contradicts our biochemical studies. It also contradicts cross-linking studies in extract. This just highlights what I've mentioned. This is one of our structures of P14 complex with SF3B1. Here's the EM structure, and you can see that the core RM structure is the same, but these helices are not, and the SF3B1 structure is not the same. So we have a picture of the elephant now, but it disagrees with the little other snapshots that we have, including a considerable amount of biochemical and other data. We've been studying the POMB deletion mutant. I'll just very briefly refer to that. The P14 deletion is viable in POMB, and it took us a long time to determine a phenotype. This just shows the clumping phenotype that occurs when the cells are growing to saturation. We solved the structure of that complex, and it's different from the other two that I showed you. So I'll just mention that this plasticity in the P14 molecule is reminiscent of a rearrangement that was seen in the U1A protein, in which blocking at the RNA binding face is relieved by rearrangement that allows it to interact with its cognate RNA. And those were structures, these were some of the earliest spliceosome protein structures that were determined by Kiyoshi Nagai's lab many years ago. So I'll just end with this photo. I was privileged to visit Kiyoshi a number of years ago, and he took me on a nice tour of Cambridge past the old labs, and we ended up at the Eagle pub where Francis Crick announced he'd solved the secret of, he and Watson had solved the secret of life. And we all really owe Kiyoshi a huge debt for the work that he put into our understanding of spliceosomal structure. And as you know, he's no longer with us. I'd just like to thank my lab, the current members shown on the left, former members and undergraduates on the right. And I'm sorry, people probably won't be coming to Alberta this summer, but here's one of the treats should you choose to do so in the future. So thanks very much.
Great. Thanks a lot, Andrew, for this interesting and in-depth tour de force of the spliceosome. Now I would be waiting for questions. And I was just wondering if while we were waiting for questions, I was curious for the work on mutant that you actually have done in yeast. I was wondering if you did or tried to look at the global change in splicing pattern with this mutation. Did you try to do like a transcriptomic analysis to see, for example, a certain group of messages that are more spliced than others and so on?
Sorry, do you mean with the PRP8 mutants?
Yeah.
No, we haven't. So the thing is that those, it's sort of interesting. None of those all of those mutants are perfectly viable, and it hasn't really been explored. But they, on normal, there's really no effect on normal splicing. And so you only see the phenotype with compromised substrates.
Okay.
In which, for example, you've mutated the splice sites or mutated the branch nucleotide, and that's when you see the rescue. One other thing which I didn't mention is that, without going into details, is one of the mutants I showed, which is I think it's an I to Y mutation in that hairpin. That's actually inconsistent with the EM structures as well. So it's perfectly viable, but in the EM structures where that hairpin is contacting anything, it's jammed up actually against the branch duplex, and that interaction is impossible in that mutant. So I don't know what that means.
Oh, interesting. I'm just trying to see if there is any other question. Yeah, so here's one from Mustafa. Is the core of spliceosome conserved and behaves as a constitutive complex splicing machinery, or it can be heterogeneous, or that in a way to trigger splicing? I guess it has to do with the dynamic. Is it a static or dynamic complex where factors come and go?
Oh, well, yeah, their factor, I presented a very simplified version, right? I mean, the core is believed to be conserved amongst all of these spliceosomes, human and yeast, but so for each of the individual steps, but there are considerable rearrangements even between the first and second steps. Both in, there are, I didn't show, but there are at least 3 different other C complexes that where factors come and go, but before the splicing reactions occur.
Okay, interesting. Here's another one. A recent manuscript that's from Anna Krichvetsky. A recent manuscript in Genome Research suggested that RNA components of the spliceosome regulate tissue and cancer-specific alternative splicing. Could you please comment on this?
That RNA, sorry, that the RNA components of the spliceosome?
The RNA components of the spliceosome regulate tissue and cancer-specific alternative splicing. I guess she's referring to regulation of alternative splicing by core components.
Yeah, I honestly don't know that much about that, so I can't really comment. Of course, with sequencing, there have been many mutations that have been identified in the protein components, which are related to various cancers, specifically SF3B1. I think people are familiar with that. There's one mutation in particular in the heat repeats, which occurs in many tumors. And actually, again, the EM structures are very beautiful, but they were not useful in elucidating the mechanism behind that. That really had to be figured out by Jim Manley's lab last year. Biochemically purifying spliceosomes and showing a link between SF3B1 and clearing of the branch region in splicing selection.
Okay. Here's another question from Christopher Jones. Can you comment on the source of the 2 structures in the crystal? For example, packing?
Oh, okay, yeah. Not really. There is some packing. The hairpin and the open form interact with other molecules, so that may be influencing things. It's actually interesting. The morphology, we could isolate crystals that just had the one form with the hairpin, but their morphology is separate. Is different from the form with 2, and so so when we wanted to look at what was happening, once we realized that we thought the 2 forms were important, we could just manually pick out the crystals that we wanted to look at, so that we would look at be able to see both forms.
There's a question here that apparently I have missed in the midst of all the questions coming in from Robert Weinberg. Is there any pausing or backtracking while the spliceosome operates, or does it act as in a continuous, straightforward manner?
I missed that, sorry.
Yeah, so this is about recursive splicing versus continuous splicing. Is there a pausing or backtracking in the spliceosome, or it's working in a straightforward manner?
I don't think it's...
Good luck with that one.
I don't think it's straightforward, right? you know, it used to be portrayed that way. Melissa Moore basically showed that through fluorescence studies that almost all of those steps are reversible. And actually, I forget where that was from. There was even a Nature paper years ago showing that the chemistry was even reversible. Now, what the biological relevance of that is, I'm not sure. I mean, it's engineered to pick the right sites and to go forward in a reversible fashion, but you know, never say in irreversible fashion, but never say never.
So here's one, I guess, last question from Andy Berglund. For the EM structure that showed P1450 angstrom from the U2 branch point duplex, was there another RNA element near P14 in the EM?
Yeah, hi Andy, thanks. No, there's no RNA nearby. And so, you know, either we've been completely wrong over many years and through many studies, or else something else is going on. And I think what it is that these are all just snapshots. The EM structures are snapshots. Obviously, an individual domain or a portion of a domain is a snapshot. What I didn't mention, and I don't think was apparent from the slide, is that that region of SF3B, that's at the N-terminus, and there is a large section of that terminus which is disordered in the is not visible in the EM structure. It's about I think a 60 amino acid gap, and so you could almost think that we could model that the P14 or that portion of SF3B may be moving around. There may be considerable dynamic movement, and I guess our fantasy is that it is an initial branch point recognition that delivers that to the core of the splicing machinery, and then we're seeing what it relaxes to. Okay, no, so there's no RNA nearby. Sorry.
So what I was saying is one very, very last question from Stephen. I can imagine Stephen Rader, I don't know. Is there any evidence that PRP8 RT domain is still active on an evolutionary time scale, i.e. 1 Rx, I think, reaction per century?
Is there any evidence that, that's not been, or certainly we haven't looked at that. All of those catalytic domain, quote, catalytic domains of PRP8 are dead, right? They've either been mutated or, yeah, they've been mutated in such a way that they don't possess any enzymatic activity in yeast and human PRP8. But I don't think anybody's necessarily looked at that that extensively.
Okay, great. Thanks a lot again, Andrew, for this exciting talk. Thanks, Andrew and Francois. And thank you all for participating in today's edition of the RNA Collaborative series. I think it's probably a fresh thing for you guys to see some talks from Canada. And I especially thank all the people who actually stay until the end. And hopefully we'll see you in the next RNA Collaborative meeting. I think it will be next month, I think. And looking forward to see you again and hear exciting talks. With that, thank you again, and see you later.
Okay, thanks very much, everyone. That was really nice.